All right, let's review cellular respiration before we talk about what happens in the electron transport chain and what happens in the process of cellular respiration when oxygen isn't available. So, we know cellular respiration, the purpose is to take glucose, the energy that we're getting from our food, and break it down into usable cellular energy in the form of ATP. Now, it's really important to remember that cellular respiration happens in both plants and animals. So, in both organisms, we're going to start with glucose. And it's going to go through the first step of cellular respiration, which is glycolysis. And the purpose of glycolysis is to break down glucose. Glycolysis is going to happen in the cytoplasm and it does not require oxygen, so it's called an anaerobic process. Out of glycolysis, we are going to get a couple molecules. We get two pyruvates, two ATPs, and two NADHs. Remember, NADH is just a high energy carrier molecule, and these NADHs are going to come in play later when we get to the electron transport chain. So after glycolysis, we focus on the pyruvates. Pyruvates cannot enter the second step of cellular respiration, the Krebs cycle. They have to be transformed into a different molecule. To do that, pyruvate is going to get the assistance of coenzyme A, and when that reaction happens, it produces two acetyl CoAs. Acetyl CoA is the molecule that we need to enter into the Krebs cycle. From this reaction, we also get out two carbon dioxides as waste, and then two more NADHs. So now our acetyl CoAs can go into the Krebs cycle. And it's important to note that this only happens in the presence of oxygen. The Krebs cycle is an aerobic process. It needs oxygen. So once these two acetyl CoAs go through the Krebs cycle, which is taking place in the mitochondrial matrix, the products we get out are four carbon dioxides, two more ATPs, and then we get three high energy carrier molecules. We have two GTPs, two FADH2s, and six NADHs. So what we're going to focus on is we're going to look at what happens next. Where do all these high energy carrier molecules go? So in your notes, you're going to have thinking question number 5. What is the point of the Krebs cycle or the citric acid cycle? The take-home point for the Krebs cycle is that the Krebs cycle is taking the energy that was originally stored in pyruvate, and that energy is then going to be carried by those high energy electrons, or our electron taxis, our NADH, our GTPs, and our FADH2s. So because of those high energy taxis, we're able to enter the electron transport chain, which is the second step of reactions in our aerobic respiration. So the electron taxi shuttles NADH, NADH, or FADH2, and GTP from the Krebs cycle and from glycolysis are going to carry the high energy electrons into the inner mitochondrial membrane. What's going to happen is the electrons are then going to be passed down a series of transport enzymes. It's like an assembly line or a series of basketball players passing a ball to each other. This is exactly like the electron transport chain in photosynthesis, except there's no photosystems. So what's happening is the electrons get transported from one enzyme to the next, and as they do that, again, they're going to be losing energy. At the very end of the electron transport chain, oxygen is going to be the final electron acceptor. I can guarantee you that that is definitely a point that you're going to need to know. A lot of test questions are going to ask you what is the final electron acceptor in the electron transport chain, and it's oxygen. And that's important because we're going to use that oxygen plus those electrons to make water. So here's a diagram of the electron transport chain. It should look pretty similar to you. You have your electron transport enzymes. The electrons are going to bounce from enzyme to enzyme. And then at the end, you have another thing that's very familiar to you, ATP synthase. So as I said, as the electrons are being passed down the chain, they're losing energy. And as they lose energy, that causes hydrogen ions to be pumped out of the inner mitochondrial membrane through hydrogen pumps. So then the hydrogen ions, There's going to be a high concentration on the outside of the membrane and a low concentration on the inside, making the outside more positive than the inside. And this creates a concentration gradient. And what we know about cells is that they like to maintain equilibrium. They want the inside and the outside of the membrane to be balanced. So to solve this imbalance, what's going to happen is the hydrogen ions are going to diffuse back in. And the movement of those hydrogen ions through the ATP synthase is going to transform a molecule of ADP into the energy that we want, ATP. And then at the end of the electron transport chain, like I was saying before, hydrogen ions, extra electrons, are going to combine with oxygen, our final electron acceptor, to make water. So here's just a diagram showing you what's happening. We have the inner membrane of our mitochondria right here. And what happens as these electrons are moving down, these hydrogens are moving out, creating that concentration gradient. So to fix that imbalance, we're going to pump the hydrogens back through using ATP synthase, and out of that process we create ATP. So in your notes, wherever it says let's label together, instead of labeling in our notes, what we're going to do is we are going to draw onto our diagram. So on your diagram, you are going to add step 3 of cellular respiration, or the second part of aerobic respiration, the electron transport chain. Remember, the electron transport chain is happening in the inner mitochondrial matrix. And what you're going to do is you're going to draw arrows from every single cell energy taxi that you have and show that those are going into the electron transport chain. In the electron transport chain, we know that oxygen is our final electron acceptor. And from those electrons and the hydrogen ions, we're going to create water as a product. Also in the electron transport chain, as those electrons move down, we're pumping H pluses out of the membrane, which creates a concentration gradient. ATP synthase, through the process of chemiosmosis, is going to pump those hydrogen ions back in the membrane and create about 32 to 34 total ATP. Now, this is a range because scientists believe every time that we go through cellular respiration, we're not exactly making the same amount. So they just give about a range of about that 32 to 34 are created from the electron transport chain. So why do we really need to breathe in oxygen? Thinking question number 6. Why is it so important that we breathe in oxygen? And the answer to this is, like I've stated, oxygen is the final electron acceptor in the electron transport chain. We need oxygen in order to make ATP, and then we're going to create water out of that reaction. So thinking question number 7, in your own words, summarize the overall point or purpose of the electron transport chain. And the purpose is, what we're doing is we started with glucose. And we took the energy that was stored in glucose and transformed it into pyruvate. And from pyruvate, we took that energy and put it into NADH, GTP, and FADH2. So we're using that energy that originally started in glucose and is now in the form of our high energy carrier molecules to create a concentration gradient, which then in turn ultimately makes ATP for the cell. So here's all the ATP that we've gotten from the process of cellular respiration. Glycolysis gave us 2, the Krebs cycle gave us 2, and the electron transport chain gave us 32 to 34 ATP, which gives us a grand total of 36 to 38 ATP for the whole process. Here's just another diagram showing you what's going into each step and what's coming out. I think our diagram's way better. And this is just another one showing you the overall big picture of what's happening. We're starting with glycolysis and breaking down glucose. We make pyruvate. We transform that pyruvate into acetyl-CoA, which then can enter the Krebs cycle or the citric acid cycle. And then those high energy carrier molecules are going to enter into the electron transport chain to create ATP. So I want you to take a second, pause the video, see if you can fill out this chart on your own. Hopefully you actually paused the video and tried to do this by yourself. Here's the correct answers. Check yourself, make sure you have them right. With cellular respiration, really the main things you need to know is where does it happen? Does it require oxygen? And what's going into each step? And what are the products? So let's think about this. If cellular respiration requires oxygen to occur, what happens if oxygen is missing? What's going to happen if oxygen is missing is our body still needs to make ATP, but it's going to go through a different set of processes known as fermentation. So without oxygen, the process of fermentation is going to occur. So what happens is the pyruvic acid from glycolysis gets converted into something different. In plants, it gets converted into ethyl alcohol and carbon dioxide. That also happens in yeast. That's how we're making bread, and it's how you make alcohol. And then in animals, it gets transformed into lactic acid. Lactic acid is what makes your muscles sore after a really hard workout. And the only ATP produced is the ATP from glycolysis, so 2 ATP. So fermentation isn't very efficient when we're talking about how much energy is produced, because each time we go through, we're only getting 2 ATP as compared to the whole process of cellular respiration, where we would be getting 34 to 36. One glucose runs out and then fermentation stops. And then it's going to recycle NADH back into NAD plus so we can repeat the cycle if needed. Here's a diagram that's just showing you the pathways that our pyruvates can take, either going through the aerobic process, going through the Krebs cycle and the electron transport chain, or the anaerobic process when oxygen isn't present, and the 2 types of fermentation and where each one of those happens. So you're going to add this to your diagram. Off of your 2 pyruvates, you are going to draw a new arrow and say that no oxygen is present. It's anaerobic, and it's going to go through the process of fermentation. You need to be familiar with the 2 types of fermentation. There's lactic acid fermentation, which happens in animals, and it produces lactic acid. And then there's alcoholic fermentation, which happens in plants and yeast. Alcoholic fermentation is going to produce ethyl alcohol and carbon dioxide. So here's a tricky question. What type of respiration do bacterial cells undergo? And why this is tricky is bacterial cells can undergo both aerobic and anaerobic respiration depending on if oxygen is present or not. So you're going to have a big chart on the very last page, and it has a column for photosynthesis, a column for cellular respiration, and then a what they share column. What I want you to do is for each one, photosynthesis and cellular respiration, see if you can list out their overall function, where they occur, what reactants go in and what products come out, and then their equation. So take a minute, pause the video, see if you can fill this out. Once you fill it out, check with me. If yours is a little different, totally fine. But know for each one these things. And then when we're looking at that middle section, what are some things that they share? What are some common processes? Some things that I can think of is they both have an electron transport chain. They both use a concentration gradient to create ATP. ATP synthase is used to create ATP. You could say they both happen in plants. And then let me know if you can think of any other things that I missed that the 2 processes have in common. All right, so that's the rest of your notes. You are going to be totally fine for your tests. You learned all the hard stuff. This was the easy stuff. Take your time, go through your note packet, do your diagram, and look over your review sheets. Happy studying!